I'm going to talk about both the brain and the retina. Back to that yellow spot a little bit. And I'm going to talk about a couple of studies that are being done presently in progress in collaboration, our group in Oregon with John Erdman's group at University of Illinois, Elizabeth Johnson at Tufts, and Abbott Nutrition, Matt Kuchen and Suzette Pereira, with funding from Abbott Nutrition through the Center for Nutrition Learning and Memory at Illinois. One of the issue that we're trying to address is whether xanthophylls promote optimal brain and retinal development. Now, many of us here have spent years or decades looking at xanthophylls in the context of aging, AMD, and as evidenced this morning, Alzheimer's disease, diseases of aging. There's a whole lot less known about the possible role of xanthophylls in development. And there are many possible mechanisms by which these compounds could affect development of the fovea in particular, where these compounds are so concentrated, and the brain. And I'm not going to go through any detail, but for example, antioxidant protection is particularly important in infants, and particularly in the infant retina, where they may be less mature, control of blood flow and oxygenation. Blue light protection is really important for infants who have incredibly clear ocular media. There's issues of cellular respiration in development. There's been suggestions or speculations that xanthophylls enhance gap junctions between neurons and glia, incredibly important in brain development, are associated with microtubules. And of course, we've heard a bunch of data here and in the past about improved visual function in adults with lutein and zeaxanthin supplementation or higher levels in the diet. Things like improved lower thresholds, enhanced visual acuity and contrast, and faster temporal processing, which Randy and Lisa have really pioneered that, which is more central function. But we know that the development of the visual cortex is experience dependent, dependent on the quality of the visual stimulus coming in. And so to the extent that there might be effects at the retinal level on development of visual acuity or contrast sensitivity, those may cascade into effects on cortical development as well. So I don't think I need to belabor the point where we're using a non-human primate model to address these questions for reasons that I think you all know, that they have a macula and a fovea, more advanced brain structure and cognition, and that they accumulate lutein and zeaxanthin in the retina and the brain, unlike other mammals. And obviously, we're able to very strictly control diet and conduct a lot of intensive evaluations. So I'm very impressed with the amount of testing that Lisa's subjects are going through. That's quite amazing. So the study that's now in progress, and I will not be able to present very much of this, just a bit of preliminary data, the later part of the talk, involves 24 newborn rhesus infants in 3 groups on a low lutein formula, high lutein formula, and we have a breastfed reference group. And we're doing 5 different modes of retinal imaging on these infants, as well as measures of cortical and retinal function, and several types of brain MRI scans. So we're going to be very excited next year presenting you a whole lot of data from this study. Meanwhile, we've done an initial pilot study with just 4 infant monkeys for 4 months of duration of feeding on a low lutein formula and a high lutein formula. And these were not from birth, but from 1 to 3 months of age. And the major purposes of this was first of all, to pilot all of these outcome measures with infants, with young infants, and particularly to confirm that this difference in lutein intake would result in differences in lutein levels in the brain. So I'm going to show you that data. So these are the serum levels from these infants, and these are the 4 individuals, 2 infants on high lutein and 2 on low lutein. And you can see that by 4 weeks of age, there's about a 3-fold difference in serum lutein, and by 16 weeks, about a 5-fold difference in serum lutein. For zeaxanthin, it took longer for levels to diverge, but by 16 weeks of age, zeaxanthin was undetectable in the serum of these infants, in the low lutein, low zeaxanthin group. And zeaxanthin was present at about 10% of the level of lutein in the formula. I forgot to mention that. This is in the livers after 4 months of feeding. There's an 8-fold difference in levels of lutein in the liver. Again, these are the individual infants. And liver beta carotene is also reduced by about half. In the brain, this is data from John Erdman's lab. We analyzed several brain regions. And in prefrontal cortex, in the low lutein infants, in both of them, lutein was undetectable in prefrontal cortex, where it was at pretty substantial levels in the 2 that were given the high lutein formula. In occipital cortex, on the other hand, there was a substantial reduction, but the presence of measurable lutein. And this next graph shows this same data for prefrontal cortex and occipital cortex, but also the 4 other brain regions. You can see that also in superior temporal cortex, lutein was non-detectable, and also in cerebellum in the low lutein group, where it was present but very much reduced also in striatum and hippocampus. So this has confirmed for us that these levels of feeding are producing really substantial differences in lutein levels in the brains. So conclusions excuse me, from the pilot study are that we got a 5-fold increase in serum lutein with the lutein supplementation, the high lutein formula, and 8-fold increase in liver lutein, dramatic differences in lutein content of all brain regions analyzed. But it's very interesting and notable that primary visual cortex showed preferential accumulation of lutein in both the high and low lutein groups. And this is something that, Liz Johnson and I also found previously in adult rhesus monkeys that were supplemented with lutein, that there was highest levels found in the primary visual cortex, occipital cortex. Now, to go into the main study, what I'm going to do is sort of step back. These are our the major outcome measures that we're doing in this study. But I want to step back and give background for the process of foveal development in particular, and the kinds of measures that we're using to measure foveal development. So, especially for those of you who are biochemists and nutritionists, the critical thing to know is that the density of cone photoreceptors is incredibly high in the center of the fovea. So whereas across most of the retina is only about 5,000 per square millimeter, there is this incredibly steep peak right at in the fovea of up to 150 to 200,000 per square millimeter. And but the infant retina is not that way. It does not have this peak in cone density. This is something that develops rapidly, primarily postnatally in both human and the monkey eye. And this little schematic shows, here, photoreceptor layer and then the bipolar and ganglion cell layer in an immature eye at or a little bit before birth. And postnatally, there are very rapid changes in the cone packing. So cone photoreceptors are moving in toward the center, packing tighter and tighter. They start out short, stubby, and fat, and as they pack inward, they get long and skinny. So the length of the outer segments of the photoreceptors gets longer and longer, and there's the formation of the foveal pit. The word fovea, of course, means pit. Changes in RPE pigmentation, and the pit is shown here, the foveal pit, with the photoreceptors here, and this illustrates how the other cell types, bipolar cells and ganglion cells, to which the photoreceptors connect, are displaced laterally. And all of this structure is designed to optimize visual acuity, to remove anything from the path of light through here that might distort the visual image, so that light has a straight path right to the photoreceptors. And another specialization of the center of the fovea is the absence of even capillaries of the network of retinal vessels. So this is what's called the foveal avascular zone in the right center of the fovea, which develops all in concert. So all of these different changes are coordinated to create this very special structure. So these are two key measures of foveal development. And this is a graph for human infants of the development of both cone density and outer segment length over the first several years. So these are continuing to develop out past 4 to 6 years of age in human infants, not quite at adult levels by even 4 to 6 years of age. And both of these parameters are closely correlated with the development of visual acuity. So outer segment length we can measure in vivo by ocular coherence tomography, and cone density, as I'll show you, we can now measure by a new technology called adaptive optics imaging. So this is the ocular coherence tomography, which gives us measures of retinal thickness. And I think you're all now pretty familiar with this. We've seen a lot of these kinds of images. This shows the kind of images we're taking from our infant monkeys, where we can look at various parameters of the structure of the fovea. So we can look at the width, the depth, the volume. We can look at the thickness here. This is a young infant. This is an adult. And these are just a couple of the parameters. This is this thickness of the foveal pit here, thickness of the retina and the foveal pit. And this is foveal width, which is reduced with maturation. So the foveal pit starts out shallow and wide and becomes narrower and deeper with development. And this is an example of the correspondence of these OCT images here. This is from a recent paper to histology. So one is able to segment these different layers and get thickness of the different cellular layers of the retina, 10 different cellular layers. So these are examples of images of a 2-week and 24-week-old infant monkey with these different layers segmented. And again, and this arrow is showing specifically the the photoreceptors from the outer segments through the outer nuclear layer, and showing this increase from 2 weeks to 24 weeks of age. So here's some developmental data that I can show at this point. So as we would expect, there is a progressive increase in the length of the inner and outer segments, and also the thickness of the outer nuclear layer, which is the photoreceptor nuclear layer. Those stack up on top of each other. There's, at the same time, a decrease in the thickness of the outer plexiform layer as the cone axons move laterally. And the inner retinal layers all are decreasing in thickness at the center, in the foveal area, again, as those connections all displace laterally. We're also measuring macular pigment optical density, and I don't have that data yet to show you, but just to say that, yes, we are measuring macular pigment optical density, and what I can say is that we see a very rapid increase at very low levels at 2 weeks of age, and a very rapid increase up to 24 weeks of age, and very typically see this kind of three-peak profile. The newest technology that we're using to look at retinal development now is adaptive optics imaging, which allows quantification of cone density in vivo. This is a newer technology that is using a technology that was developed for astronomy to allow good visualization of stars and galaxies. And what it does is it uses a computer deformable mirror to compensate for optical aberrations in the image. And it does this so perfectly that in a living eye, it is possible to visualize individual cone photoreceptors. These little white dots, if you can see them, are individual cone photoreceptors. And so one can get a measure of this longitudinally in vivo. Here's the process. We take each image is a 4 by 4 degree tile or frame. Each of those is an average, actually, of 40 frames. These are montaged. We take about 30 of these individual tiles, montage them together, and then there's a lot of computer post-processing to identify the cones and to generate a heat map of cone density. This is for an adult monkey, and you'll see that the cone density increases. The red is the highest, and then there's a crater. The reason is that although this technology has incredible resolution power, it's not good enough to resolve the center of the adult fovea, where the cone density is 150 to 200 cells per millimeter square. We are able, in our monkey infants, to resolve up to about 50 to 100,000 cells per millimeter square. So this is an example from a 2-week-old monkey infant, 1 frame, and I've blown up one piece of that so that you can more clearly see the individual cones in their very characteristic hexagonal array. And then this false color map gives density. The center of the fovea is about here in this image. This is just the individual foveal frame, not montaged yet. And here's an example of an increase from 1 to 2 months of age in a monkey infant in cone density. So we're generating this data as we speak. Just got a bunch more this morning. So we're basically waiting to see until what age we can resolve cone density. At some point, it's going to be too high, and we'll get the crater as is seen in adults. but it looks like we can resolve cone density in these infants at least out to 3 months of age. So I'm going to skip through this because I'm running out of time. We're doing some I just mentioned very briefly, measures of macular cone function with the multifocal retinogram, which gives a map of retinal function across the macula. This is infant responses, but I'll skip over this. The other thing we're doing is a number of types of brain imaging, both structural MRI, diffusion tensor imaging to look at the development of fiber tracts, and also resting state functional connectivity MRI. 2 years ago, I presented some data with this method showing really dramatic effects in adult monkeys that had been on different levels of omega-3 fatty acids their whole lives. We're, of course, really interested to look developmentally at effects of lutein on this, this sort of measure, which looks at the correlation between activity in different cortical areas as a measure of their functional connectivity. It also gives measures of the modular organization of the cortex. So finally, what we conclude at this point is that formula lutein content clearly impacts brain lutein in primate infants, that there's a preferential accumulation in the primary visual cortex in these infants, that we can implement multiple measures of foveal and brain development in these infants to be able to measure in a quantitative way and in ways that have not been done before in either monkey or human infants, several different aspects of development of the fovea postnatally. And we feel that these monkey infants provide us a powerful model for studying early carotenoid nutrition. Thanks.
Dr. Neuringer, so we have time for a few questions. Julie?
Well, that's fascinating. I'm wondering if you can speculate or hypothesize how you think the OCTs in adult beings would be in people fed lutein versus not, based on early changes you're seeing.
I'm sorry, you're saying in adults?
So based on what you're seeing, thank you. Based on what you're seeing with the couple of monkey infants and the couple of humans, in terms of OCT changes with development in lutein compared to no lutein, can you speculate on what might be lasting changes that might last through until adulthood?
Right, and first I have to say we don't yet, what I showed you was just developmental data. We don't, I mean, this study, the main study is still masked.
Yes.
So we don't know, we have
Yes,
no idea yet on whether there is an effect of lutein on those parameters. That's what we in terms of whether, though, there would be just, if there's an effect, would it possible it could be a delay in development, a slower trajectory. But
so ultimately a lower, you're hypothesizing a lower cone density perhaps?
Right, right. So that there is, I can say there is very great variability from the not gigantic amount of data that's available, and this is mostly actually from histology rather than from the adaptive optics imaging yet. But there's a huge variability in normal adults in foveal cone density, threefold. You know, there's some people who have like what I call pilot vision, you know, who are like, and people like me who are, you know, not so great. But so, you know, how much of that could be affected by early nutrients? It's an open question. We just don't know.
It's interesting thinking about the cone density in relation to carotenoids that Richard Bone observed. We observed in Women's Health Initiative subjects that of the 3/4 that could remember enough to report, the 25% who were formula fed at birth compared to just having had breast milk, or in that time, formula feeding was cow's milk, basically, formulas.
Missing a lot of things.
Yeah, a lot of stuff. Had lower macular pigment. And so we do wonder whether there's a lower cone
Right,
density.
right, and I saw that poster yesterday showing that macular pigment actually related to, at least self-reported, early vegetable intake better than it did current vegetable intake. So whether that could be set at an early time is, yeah, incredibly interesting question.
Hi, Martha. Thank you for
Hi,
the great
Jim.
talk. A couple of questions. How long do rhesus monkeys nurse in the wild? You mentioned a 4-month formula feeding.
Yeah, it's, there isn't like a day of weaning like there is in rats. It's a really long, slow process. So they are almost exclusively breastfeeding up to about 2 months of age, but they're already starting to, check out other foods.
I figured it'd be quicker than
So,
a human.
and I'd say, most of their intake up to 4 to 6 months, and then.
Yeah, okay, great. And then I'm also interested, you said you had some pilot data on the macular pigment increase, and you said it was pretty dramatic early on. Can you characterize that just very basically?
And
Is it-
I'm not ready to present that data yet, but,
Okay,
but
okay.
I can say it's really low at 2 weeks, and it
It
seems like there's a
increases
pretty
rapidly.
fast linear increase, and we have only a couple of measurements out to 24 weeks of age at this point.
Okay. And lastly, we've been talking a lot about the localization of function in different neural tissues, brain, retina. And it strikes me in seeing a lot of the data, you showed occipital accumulations being very high and certainly in frontal lobe. It strikes me that, including the retina, that we might just be seeing, a relationship between the deposition of, in particular here of interest, the macular carotenoids, in areas that maintain incredibly high metabolism and the concomitant oxygen tension. So could you comment on that?
Yeah, no, that's a very reasonable and interesting hypothesis, and across brain regions, whether you could correlate that would be interesting. We're going to be looking at many different specific brain regions. So,
Excited to see what happens. Thank you.
So Martha, the imaging data that you were showing is just incredibly cool. I wanted to know if you all were collecting any behavioral data to go along with those data. And I'm asking actually, I think for a similar reason that Jim did. One thing we see in adults is relationships between macular pigment, visual function, visual motor function, and motor function independently of those other two. And when I saw a complete lack of detectable lutein and cerebellum, I immediately started thinking of motor development. So I was curious to know if you're collecting any of that information.
yeah, this study does not include behavioral outcomes. That's obviously something we're extremely interested in. And I have to say, the thing is, these infants are having a pretty intensive series of sedated procedures to do all of this imaging,
Right.
which, from my experience actually with some other studies that tried to do it all, it's really not compatible. You've got to sort of do one or the other, So yeah, I'm hoping in the future we'll really get into the behavior, including motor and a number of cognitive measures.
We have time for one more question.
I have one
Will?
quick question. I'm curious about your resting state paradigm that you're using.
Yeah.
Could you describe how the monkeys are set in the MRI? Are they anesthetized? Or
Yes,
are they-
absolutely. They're
we
1% isoflurane. So we did a pilot study, and this is, and I didn't acknowledge them, but this is with Damien Fair, who's an expert at OHSU on resting state connectivity, fMRI in particular. He's actually done a lot of work on normative development in humans of the resting state parameters and compared them to children with autism and ADHD and see how they diverge. So when we did our first monkey work, we did a pilot study looking at different anesthetic regimens and chose 1% isoflurane as the best for the resting state functional connectivity data. I mean, they can't be, you know, awake and still in a magnet for.
Right. I'm currently slugging through some resting data myself in humans, and so I was curious of what state they're in and how we might be able to compare those.
Right.
So thank you.
Yeah. Yeah.
So thank you, Dr. Neuringer. Our